Today I'm going to be talking about developmental biology, and we're going to be discussing about the first third of what I'm planning to teach. So, to start off with, what is developmental biology? It's a study of how organisms evolve over time and how they create their organs and stuff like that. So, there's going to be a couple things that influence that, and so that's going to be differentiation up here. That's going to be when cells go into different categories, when they differentiate and they have different functions. Patterning, which is the organs you have and the limbs you have on your body. Morphogenesis, that's again the function and getting your organs and your limbs. Growth, just the growth of an organism. Reproduction, regeneration. Environmental integration, how you integrate with the environment. And evolution. So those are some of the things that developmental biology is going to talk about. So when we're discussing developmental biology, it's important to ask how we coming to our conclusions, where we're getting our data from. And so that's going to be determined by you having organisms and doing tests on them, doing research on them. So here are some of the model organisms and what you would look for in a model organism. So you need a good size. You need a good generation time. If it takes too long, you won't be able to see how things influence the development of the body. You need to know how accessible the embryo is, because if the embryo is not very accessible, you won't be able to do studies on it. It needs to have an accessible genome. You need to know what the genome is, or that's a factor when you're considering what organism to use. You want to look at how far away it is on the evolution area lineage to see how much you could be, how many things you could have in common with that organism, how easily you can manipulate the DNA, and then money concerns, obviously. So I guess the first thing we should start with is how does two cells get together and form the embryo? So that's going to be called embryogenesis, and that's what we're going to talk about over here. So I'm just using a diagram of a female reproductive tract in humans, but these similar stages occur for all species. So when fertilization occurs, it's obviously when the sperm comes into contact with the egg, and so the sperm will actually come up to the huge cell of the egg and release the DNA into it. And so that step is called fertilization. And now you have just the egg, an egg with all the DNA for the organism, but it's going to start to go through cleavage, in which it's going to divide. And so the embryo is inside of a sphere that's called the zona pellucida, which is going to keep it the same size, but the two cells on the inside are going to start dividing. And so this is now called a blastula, and cleavage will continue for a while until we're going to get the 3 different germ layers, which will, in a lot of animal species, which will come to make up your endoderm, mesoderm, and ectoderm. And that's going to give rise to different parts of your body. So now we're going to go through implementation, which, at least in mammals, is when the egg is going to attach to the outside of the uterus, and it's going to go through some more germ layer development. And then there's a gastrulation phase, and this is when the 3 sides of the 3 layers of the embryo are going to form that we just talked about. So I won't go too much more into that, but then there's some other stages that occur, but for this video I'm just going to cover those. So now it's time to talk about patterning, and gives rise to the different organisms and their different body types. So differentiation is going to play a major role in this, and that's how cells are going to come to be different types of cells, like we just talked about. So when a cell is differentiated, it means it cannot turn into a different cell. And so a cell can become specified in which it has certain signals coming into the cell telling it should become the certain cell type. But if you put that in a different environment, it can still switch back. So that it's called you're reliably specified. If you're specified, you're going to turn into the cell. If you're irreversibly specified, you're going to turn into that cell no matter what. So maybe you'll be a nerve cell, and you're not currently a nerve cell, or the cell's not currently a nerve cell, but it's going to evolve, or it's going to transform into the differentiated form of the neuron cell. So there's 3 different types of specification that can occur. And so autonomous is a self-grounded or self-generated specification. So this comes from inside the cell, like the cell's machinery and stuff like that. The different biomolecules in there is going to determine what it's going to be. You can be conditionally specified, or the specification can happen conditionally, where the signals, the cell-to-cell contact, and the environment is going to determine what cell comes to. And then there's something called cynical, which is inside of insects, in which you have a bunch of different nuclei inside of the egg, and when the egg goes through cleavage, the cells will actually become the cells that the proper cell type, just based on the nuclei inside of the cells now. So I talked about conditional specification, but how does that occur? What are some of the signals? And so a main signal is called a morphogen, and this is a specific molecule that the concentration is going to determine the cell types of the cells around. So I drew this image here, and you can see at the more concentrated area of the morphogen, it'll be a different cell type. At the least concentrated, it'll be a different cell type. So these morphogens are normally happen through the paracrine, in which they're inside the bloodstream and they just go. The inducer makes the cell and it sends it off, and the cell that receives the signal is called the responder. And so there's 3 major types of morphogens: FGF, hedgehog morphogens, and Wnt morphogens, but I'm not going to be covering those right now. So the FGF morphogens are just involved in segmentation, which is how different segments form in the organism, and hedgehog genes are called hedgehog genes. And yeah,